Hi, this is Madeline Kolkol. It's Sunday, November 24th. This is for the lab bacteria identification motility testing. I have here my developed tubes of E. coli and S. epidermidis, and I was a little skeptical at first when I had done these. When I was puncturing the agar, I thought to myself, there's no way that anything was going to grow. It's so thick, it's so opaque. If anything, there would be just growth on the top. And in both cases, yes, there was growth on the top. But what I did find, and it's really kind of cool, in E. coli, and I don't think that the camera's really going to capture it very well, but in E. coli, from here, from the top down all the way about 3/4 almost to the tube, to the bottom of the tube, there was growth. And every angle that I keep looking at it, I do see a difference, or I do see different sides of it and exactly how far the growth extended. I probably punctured, I should have made a label of actually how far down I punctured, but maybe 1/4 of an inch into the medium. And then I have growth all the way, probably about another 1/4 of an inch down through it, and not just in what would appear to be the spiral of what was inserted, but on its own it moved, and that was with the E. coli. Of course, on the top there was quite a bit of growth. I wasn't surprised by that. I thought it was expected. However, with the S. epidermidis, there still was growth. However, it wasn't quite as distinct and formed in comparison to E. coli. Of course, the growth on the top, I think E. coli really did well in this one. But it almost has like a string-like or web-like growth pattern on the inside. It's very faint, but I found that very interesting to see in this experiment. So some of the things that I found interesting in this experiment, other than just seeing the growth and the motility testing, and to see that the bacteria not only grows but has the ability to travel within the medium, was cool. Especially learning about the different types of movement that the bacteria could have, flagellar, spirochetal, and gliding movement. One thing I'm going to do for my own personal interest is to research the gliding movement and see what kind of mechanisms that would be, because I would think it would be some sort of chemical reaction that would cause the bacteria to jump or move, maybe something more along the lines of a chemistry reaction rather than a physical moving reaction, as you see in spirochetal and flagellar movements. Definitely the kind of medium that's used would have an effect on the way that it grows and to the extent that the bacterial culture grows. When we cultured in broth, the broth is thin. It's very thin. It's watery. So I imagine that it wouldn't be difficult for the bacteria to not only proliferate, but fill every space of the broth and utilize the broth to its full potential with minimal resistance. However, with the agar, it's going to cost and use a little bit more energy for the bacteria to move around, so I didn't expect to see as much growth. Same thing with the plates. The agar port on the plates, I feel like the bacteria moved. Well, I couldn't say that it moved because the way that we set the plates up, we covered the plates with the bacteria for it to be cultured, but it appeared that it very easily grew in a horizontal motion or horizontal fashion across the plate without any resistance, but it didn't necessarily puncture the agar. In this case, it wasn't puncturing anything, but using the initial puncture, inserting the sample into the agar, you could actually see it grow downward and grow outwards from where it was initially placed. So that was my long-winded try explanation of putting my thoughts into words. So that is it, and I've got glasses this week because somehow I scratched my cornea. So doctor's appointment tomorrow, and hope for the best. We'll see you next week.